In this lecture, we will discuss the development of the respiratory system, and as we finish off that discussion, we'll also consider how the pleural, pericardial, and peritoneal cavities form. We want to be able to start with the primitive gut tube, show how the respiratory diverticulum develops from that gut tube, and then forms the trachea, bronchi, and lungs. We're going to talk a little bit about the origin of the cells that develop into the lung tissue, and we're going to compare and contrast the development of the different stages of lung development. As I said, we'll also then get into the subdivisions of the body cavity as it develops into pleural, pericardial, and peritoneal cavities. The development of the lung begins at 4 weeks, and the generality is that the epithelium of the respiratory system begins from endoderm, while the connective tissue components, the cartilage, the smooth muscle, the rest of the connective tissue, develops from splanchnic mesoderm. We're going to talk about the development of the trachea, the bronchi, and the lungs. We'll consider lung maturation, and then talk about the development of the body cavity. If we look at this cartoon on the right, we can see the foregut in yellow, the midgut in green and blue, and the hindgut in purple. At 26 days of development, a small diverticulum appears in the foregut. It's anterior in direction, and as it develops, it's going to develop a little bit of an evagination, and this will be called the laryngeal tracheal diverticulum. This is another way to look at it. This is the foregut, and we see there begins to be a diverticulum, or the lung bud, that begins to develop. And as that lung bud grows downward, a septum is going to develop between the lung bud and the rest of the foregut. And that septum is going to continue separating the developing trachea from the developing esophagus. So we'll call this septum the tracheo-esophageal septum. We can ask questions about how this subdivision started and what are some of the molecular signals that help to give rise to the esophagus posteriorly and the lung bud ventrally or anteriorly. And we know that several of the factors involved in this initial development of these structures are fibroblast growth factor, Wnt, and bone morphogenetic protein. These form gradients that influence NKX2, which is important for the formation of the lung bud. One of the factors that regulates FGF10 is retinoic acid, which when it increases the levels of FGF10. At the same time, in the region of the notochord, we have noggin being expressed in the splanchnic mesoderm, and that's going to form another gradient and help influence the expression of SOX2, which is a prominent molecular signal found in the esophagus. So we can see that there's a differential expression of genes in these two regions of the developing foregut. We know that some of these players are important, things like sonic hedgehog, retinoic acid receptors, FGF10, SOX, NKX2, the bone morphogenetic proteins, noggin, and Wnt. We know these are important because in different knockout situations where these have been disrupted, the result has been either the formation of tracheoesophageal fistulas or esophageal atresia. In tracheoesophageal fistulas, communication remains between the developing trachea and the esophagus. In an esophageal atresia, there is actually the ending of the esophagus. It ends as a blind pouch, so that just the trachea remains. When that occurs, what ends up happening is that anything taken into the oral cavity would go down into the trachea. So these all play some sort of role in that initial formation of the lung bud. As the lung bud begins to develop and is separated from the esophagus, one of the things that happens is it begins to divide. And the lungs will end up ventrally, the esophagus will end up dorsally. In the region of the larynx, the epithelium from the foregut, is the endoderm, is going to form all the laryngeal epithelium, whereas the connective tissue, the cartilage, and they develop from the splanchnic mesoderm. Some of the cartilages develop from neural crest cells. So the neural crest can contribute to some of the cartilaginous structures found in the larynx. As we move down to the trachea, again we note that there is splanchnic mesoderm and endoderm, and then the lumen of the trachea. So the epithelium and the glands of the trachea develop from the endoderm, whereas the rest of the structures of the trachea, including the C-shaped cartilages and the smooth muscle, develops from splanchnic mesoderm. And that's what we're trying to depict in this little cartoon. Now we want to go back and look at this particular depiction of the endoderm and splanchnic mesoderm and make the following point. Once again, we see that there are epithelial mesenchymal interactions that are necessary for the development of the structure. Here we see the epithelium in yellow and the mesoderm in red, and one of the things that we can see are there going to be interactions between these two that are important for the development of the trachea. Again, FGF10 is important, and FGF2 is important, and it is expressed in the the endoderm. One of the things we know that in FGF10 deficient mice, lung buds fail to form. So FGF is critical for the formation of the lungs. So as the lung bud develops, it's going to divide first of all into two lung buds, and then it will continue to divide differently on the right and the left side. The right then dividing into three main bronchi, and the left into two main bronchi, much as you've seen in gross anatomy. And one of the questions has to do with this branching pattern and what regulates the branching of the lung bud. The bronchi are going to continue to divide, and they'll continue to divide until there have been about 17 generations of divisions. After birth, these will continue to divide in terms of the alveoli continuing to get smaller. And one of the things that is found is that in terms of lung development, the more cephalic portions of the lungs, those that are higher, are going to develop before the more caudal parts of the lungs, or the lower lobes. When we talk about lung maturation, we can talk about three stages. The first stage we've been talking about is the development of the lung bud. But then we can talk about, and that can be called the embryonic stage. Then we can talk about a pseudoglandular period, which goes from that embryonic period 5 weeks to about 16 weeks, then a canalicular period, and then finally a terminal sac or saccular period. There are a variety of ways that this is described, and this is the scheme that we'll use for this lecture. So that basically early on we have the formation of the lung buds during the embryonic period, and then a pseudoglandular period, a canalicular period, a saccular period, and then the final maturation of the lungs is from the late fetal period through childhood. The pseudoglandular period is an important period when the branching of the respiratory tree occurs, so that there is a branching process that takes place, and it's very consistent in the way these this branching occurs. And it continues to occur until the terminal bronchioles are formed. So all the way through the formation of the conducting portion of the respiratory tree is formed by this branching that occurs up to about 16 weeks of gestation. So those elements of the lung are developed, except those that are important in gaseous exchange. Now, when we talk about this branching morphogenesis, what we see is a process that really takes 4 steps. First, the bud is going to elongate. We see that from this step to this step. Then that elongation stops, and here we see some of the interaction between the fibroblast growth factor 10 and fibroblast growth factor 2. And then what's going to happen is the tips of that lung bud are going to widen, and finally they are going to bifurcate. And it's not really well worked out as to what are the factors that influence these different stages, especially the widening of the lung bud and then its bifurcation. It is known that retinoic acid is important with its highest levels proximally. We know that one of the things that retinoic acid can do is inhibit the FGF10. We also know that sonic hedgehog can promote bone morphogenetic protein 4, which inhibits fibroblast growth factor 10. In addition, the bone morphogenetic proteins are going to promote the proliferation of the mesoderm, and that may have a role in forming the widened tip of that lung bud. But it's not really clear what the BMPs are regulating and how they are working at a cellular level. During the next 2 periods, the canalicular period and the terminal sac or saccular period, the changes that occur have to do with the formation of the respiratory portion of the respiratory tree. So what's going to happen is that the lumen of the bronchi and bronchioles are going to become large relative to the tissues, and the tissues are going to thin out. The bronchial tree branches become narrower, and respiratory bronchioles and alveolar ducts are going to develop. That is, we're going to get a respiratory epithelium forming. In addition, the other important part that takes place during this early development is that the tissue has to become more vascular, and the capillary beds have to become closer in proximity to the epithelium. So here we're looking at that canalicular period. You can see these divisions of the respiratory tree. They are surrounded by a simple cuboidal epithelium. The blood vessels, which you can see here and here, are not that close to that epithelium. So at this point, there is really no possibility of gaseous exchange. However, as the canalicular period continues, we will see 2 changes. 1, the thinning out of this epithelium, and 2, the movement of these blood vessels closer to those flattened out type 1 alveolar cells. And as that happens, we will then see the terminal sac period occur, in which that process will then continue on at a substantial rate. that is the thinning out of the epithelium and the close proximity of the newly formed capillary beds to the respiratory epithelium. And that terminal sac period is going to continue until birth. Now, the question is, what happens that allows for this switch from the pseudoglandular period to the canalicular period and then to the terminal sac stage? So that what causes the alveolar sacs to begin to form? In addition, what happens that allows for the blood vessels to proliferate rapidly and become associated with the alveolar cells? And those signals are not well known at this time. During the terminal sac period, the epithelium is going to become very thin for some of the cells, and so type 1 alveolar cells will form. And the capillaries are going to bulge into those alveoli, and so we're going to have the formation of the capillary epithelial boundary. And so we get type 1 alveolar cells developing, a capillary network developing, and gaseous exchange can occur at this point. So from about 25 or 26 weeks on, it is possible for the fetus to survive if born prematurely. In terms of the formation of the alveoli, some of the factors that are important are things like PDGF, platelet-derived growth factor, fibroblast growth factor, and again, FGF2 and 18 are important for this later alveolar development. And retinoic acid is important, both in high and low levels. So if there's too much retinoic acid, it can disrupt lung development. And in addition, if the levels are too low, that can also cause abnormalities in lung development. In terms of the cells of the epithelium, what we see are there are these multipotential cells that form from the endoderm. And these multipotential cells can then divide and differentiate into the bronchiolar cells or alveolar cells. The bronchiolar cells are seen more proximally, the alveolar cells distally. The bronchiolar cells will then divide into two populations, the neuroendocrine cells and the non-neuroendocrine cells. And from the non-neuroendocrine stem cells, the ciliated cells and the goblet cells are going to form. And we do know some of the factors that can help drive this process, but we're not going to go into that in this lecture. At the same time, if we look at the alveolar cells, we can see that they will then develop into type 2 cells. Those cells that produce surfactant. And they can also develop into type 1 cells, but it's not certain whether these type 1 cells can come from the alveolar cells, from type 2 cells, or from both. So we'll leave that as a question mark for future study. In addition, as we said, there's a formation of blood vessels. And the blood vessels are forming by two processes. Angiogenesis, where new blood vessels are going to give rise are going to arise from pre-existing blood vessels, and also vasculogenesis, where new blood vessels are going to form from endothelial cells that come from angioblasts that come from splanchnic mesoderm. VEGF is important in regulating this process along with ephren B2 and B4. So these are some of the factors which are important, VEGF in particular, in forming new blood vessels. By 20 weeks, the type 2 alveolar cells are going to begin forming and begin producing surfactant, but only they'll begin to. So it's not until you have enough of these cells at about 26 to 28 weeks that the fetus will be producing enough surfactant to survive. Basically, the fetus needs to weigh about 1,000 grams and be between 26 and 28 weeks to survive, so that there's enough surfactant and enough capillaries are present close enough to the epithelium to permit the survival of the fetus. However, we do know that some fetuses born prior to this stage actually have been known to survive, and with artificial surfactant and other interventions, some fetuses younger than 26 weeks have made it through. The alveolar period is that later period where the respiratory bronchioles become terminal sacs and alveolar ducts and alveoli, and so these will continue to develop as the alveolar sacs subdivide from the 3rd to the 8th year. And this epithelium is going to then form where you see these very nice type 1 alveolar cells, type 2 alveolar cells here. And we're also going to want to remember that cells like this one, which is probably a macrophage, are going to develop. And that brings us to the question, where do these macrophages develop from? And they come from monocytes. So as you learned previously, the monocytes give rise to or arise from cells in the bone marrow. And these monocytes are going to circulate in the blood for about 12 to 100 hours and then enter into the lung tissue. And once they enter the lung tissue, they will become the dust cells or alveolar macrophages. At birth, the fluid that fills the lungs is replaced by air, and the fluid is cleared from the newborn via the mouth, and also the pulmonary capillaries and lymphatics are going to drain the fluid from the lungs. In terms of where the lungs are developing, they're developing in initially a body cavity, the body cavity, which then gets subdivided into pleural cavities, pericardial cavity, and the peritoneal cavity. Here we can look at lateral body folding to see, again, the formation as we see here, to here, to here, to here, and here, the formation of the body cavities. And these body cavities are going to be in communication, except at this little area here where we have the vitelline ducts. But otherwise, the body cavities are together and form one large body cavity. This is a cross-section of an embryo, the neural tube, the neural crest cells, the somites. Here's the foregut. This happens to be in the thorax, so we have the heart. The lungs are developing on either side of the foregut, and here you can see the body cavity now beginning to be separated from the pericardial cavity that surrounds the heart by the pleuroperitoneal folds. So that initially we're going to have a large pericardial cavity, a large peritoneal cavity, and what are called the left and right pericardio-peritoneal canals. They will then connect the pericardial cavity to the peritoneal cavity. If we look at the heart, then we can see that it is surrounded by the pericardial cavity. You can see that caudal to the heart, you have this developing tissue here that's going to be the septum transversum. So the septum transversum is going to be found just inferior to the heart, and the pericardio-peritoneal canals are going to be found just posterior to the septum transversum. So the pericardio-peritoneal canal, located lateral to the foregut, is going to communicate, or is a form of communication between the pericardial cavity and the peritoneal cavity. As the lung bud grows, then, as we see here, it is going to then encroach in this part of the pericardial cavity, and a ridge is going to form in this particular region, and this ridge is going to grow towards the foregut region, and it is going to be called the pleural pericardial fold, and it will separate the definitive pericardial cavity from the pleural cavity. In this fold, there's going to be one of the common cardinal veins, as well as the phrenic nerve. The pleural pericardial membranes are going to, or folds, are going to separate the pericardial cavity from the pleural cavity. As we said, they contain the common cardinal veins, which will drain into the heart, and the internal part of that pleural pericardial membrane is going to become the fibrous pericardium. And if you remember from gross anatomy, that's where you saw the phrenic nerve, also found in that pleural pericardial fold. So again, you can see the pleural pericardial membranes are going to separate the pleural cavity from the pericardial cavity and contain the common cardinal veins and the phrenic nerve. The rest of the separation between the pleural cavity and the peritoneal cavity takes place in the region behind septum transversum. So septum transversum essentially separates the pericardial cavity that's formed from the peritoneal cavity. But the lungs are in the pleural cavities, and they're going to be separated from the peritoneal cavity by the pleural peritoneal membranes and three other contributions, which all four structures are going to contribute to the diaphragm. So basically, from the posterior body wall, you're going to get these pleural peritoneal membranes. From the region of the foregut, you're going to get some growth of the mesentery called esophageal mesenteries. From the lateral body wall, you're going to get lateral body wall mesoderm growing inward. And finally, the septum transversum is going to contribute to closing off this pericardio-peritoneal canal. So that when you look at the diaphragm and see what it forms from, you'll see that it forms from septum transversum, the pleural peritoneal membrane, just right here, it's going to develop, the dorsal mesentery of the esophagus, right here, and the lateral body wall. And this tissue will give rise to the muscle cells of the diaphragm. That is, the cervical somite myotomes are going to migrate downward from C3, 4, and 5, and they'll migrate into the diaphragm. So that when the septum transversum originally begins to form, it's opposite C3, 4, and 5 cervical somites. And as we get the head folding occurring, it moves downward or caudally, and the myoblasts are going to migrate down from these somites, and the nerves are going to follow down, follow those myoblasts, and those anterior horn cells are going to give rise to the neurons of the phrenic nerve, which you remember arise from spinal cord segments C3, C4, and C5. And as the fetus continues to grow, the diaphragm will migrate caudally, and eventually it will come to lie opposite the bottom of the thoracic cavity.